So as I mentioned in the previous lecture segment, the oxidation of glucose is really the process by which the glucose, C6H12O6, will be broken down fundamentally into a waste product, which is carbon dioxide. This is called a combustion reaction. But it will happen in small steps so that the energy from the bonds, as they're being broken, can be, at least to some extent, recaptured. Now, glucose has enough energy in it equal to about 100 molecules of ATP. So one glucose equals about 100 molecules of ATP. So it's a pretty good approximation. But you'll remember in the summary equation, we can only hope to recapture or to phosphorylate ADP to an extent of about 36. So you're really only getting about 36% efficiency from this reaction. And it's because every step, there's so many different steps, and remember that in every step a little bit of energy is lost or released into the universe, and so in the end you get only about 36 ATPs. But the oxidation of glucose is an important part of it, and but we don't want to forget the phosphorylation of ADP to ATP. So let me get my pen. So the breakdown of glucose is part of it, but also the phosphorylation. Phosphorylation is also an important part of this, and you'll see it woven in throughout all these stages. Most of your ATP will be seen in the fourth stage here. So the oxidation of glucose is actually just these first three stages, to be honest. So glycolysis, pyruvate oxidation, and Krebs cycle are sort of the phases or the stages of oxidation of glucose, and then most of your phosphorylation events happen in this fourth stage called electron transport chemiosmosis. So the first stage is called glycolysis. The word glycolysis means the splitting of sugar. You're probably used to this prefix glyco, referring to something having to do with carbohydrate, in this case particularly sugar, in particular typically glucose. And lysis means to split or break. So the actual literal breaking of glucose into two pieces. This is a 10-step biochemical pathway. That means there are 10 enzymes involved. You start with one glucose at the very beginning of those 10 steps. You end with two molecules called pyruvate at the very end of the 10 steps. Glucose has 6 carbons. Each pyruvate has 3 carbons. And so all the carbons from the original glucose are still accounted for by the end of glycolysis. Glycolysis occurs in the cytoplasm, specifically, I should say, the cytosol. That means that glycolysis can happen in bacterial cells and in eukaryotic cells, or in prokaryotes and eukaryotes alike. And so this pathway is believed to be very ancient, since bacteria evolved prior to eukaryotic cells. Ultimately, now this is a typo, ultimately you get a net production of, this should say, 2 ATP. I need to get my other mouse pad. This one is not doing that well. Let's see if this gets any better. All right, 2 ATP molecules is your net ATP yield from what we call substrate level phosphorylation. And anytime we talk about phosphorylation, that means that a phosphate group has been added to something. In this case, two phosphate groups were added to 2 ADP molecules, and that gives you 2 ATP. And then we also have 2 NADH are produced by the reduction of NAD+, really 2 NAD+. And so 2 NAD+, each of those two picked up 2 electrons and a hydrogen ion to become 2 NADH. So you've got some of those electron carriers involved here. This is the detailed view of glycolysis. Shown here on the left is your reactant glucose. It gets converted by different enzymes, hexokinase into glucose 6-phosphate, phosphoglucose isomerase converts that into fructose 6-phosphate, and so on. You do not need to memorize the names of these enzymes, but that is coming for you in your biochemistry course. So you don't need to memorize those now. You also don't need to memorize these names of these intermediates, and you don't have to be able to draw the intermediates, but that's coming for you someday if you're a bio major. But what's important here is that the glucose, you should still recognize that, and that's your starting reactant. And the first 5 steps of the pathway sometimes are called the energy investment phase. If you think about a business, typically you have to put in an investment before you can get a profit. And so you have to put it in terms of respiration. Since we're trying to get ATP, you actually have to use up some ATP at the very beginning of cellular respiration before you can get the ultimate yield of ATP at the very end, which is what we said, about 36 ATP as a yield. So what's going to happen here is there's going to be, now this mouse pad is even worse. There's going to be some ATP you'll see coming in to this stage. So 2 ATP are actually invested in or required in the very first stage of glycolysis, and that's why it's called the energy investment. In the next stage, though, and in this picture it goes, it kind of doesn't make sense, but you have to start over here and then go down. In this one, you go to the final endpoint, which is pyruvate. So this is the 10th step here. So your final product of glycolysis is called pyruvate. It's a 3-carbon molecule, and remember, you would have 2 of these. So this picture is only showing one of them being produced, so everything would be multiplied by 2. And what you see here is that you get some ATP production, and you would multiply each of these by 2. So your net means when you subtract your investment was 2 ATP, your net yield of ATP is 4. So when you take out your investment, your net yield is, your investment is 2 ATP. Your gross yield of ATP is 4. But when you subtract your investment, your net yield is 2. So net means after you subtract your investment. All right, so importantly, though, you end up with 2 NADH, 2 reduced electron carriers, and 2 ATP, that's your net yield. And you get 2 pyruvate. So 2 pyruvate, 2 NADH, 2 ATP. That's what you do need to know, the products. So you start with 1 glucose, all right, and you end up with, and you also start with 2 NAD pluses and 2 ADPs. But at the end, you have 2 pyruvates, which represents the glucose broken apart, and then 2 NADH and 2 ATP. And I'm going to have a video that's going to summarize all of these things also for you. But you don't need to memorize all the 10 steps, but the concept of the energy investment, putting ATP in to get the process started, to really energize, literally energize the intermediate molecules to be able to break apart that glucose, is an important concept of glycolysis. The next stage is called pyruvate oxidation. It's really only one enzymatic step, but there's a couple things that happen all simultaneously in this one step. And this step only happens it happens if there's oxygen. So if it's aerobic respiration, then this step will occur following glycolysis. And if it's anaerobic respiration, it will happen. But this step does not happen in fermentation. So pyruvate oxidation is only in aerobic or anaerobic respiration. Now we're considering primarily aerobic respiration. So that's why I wrote here in the presence of oxygen. So assuming there's oxygen, then the pyruvate, which remember we had 2 of those in the last step, is going to be oxidized. Now this step occurs in the mitochondria, and there's an enzyme called pyruvate dehydrogenase that catalyzes this reaction. Pyruvate dehydrogenase. And this is going to also be possible in prokaryotes, but in prokaryotes, since they don't have mitochondria, this would have to happen on the inside of the plasma membrane, but the enzyme is embedded in the plasma membrane for prokaryotes. So this is pyruvate oxidation, or the oxidation of pyruvate. Here's pyruvate, shown here, step 1. There's going to be a decarboxylation. So decarboxylation means the removal of a carboxyl group. And a carboxyl group in the ionized form is C double bond O and then O minus. And when that group is broken off of the other part of the structure, this 2-carbon structure here, then this carboxyl group becomes carbon dioxide. So you can kind of see how they shaded it in purple here. So carbon dioxide is released when it is broken off of a molecule. When it's on the molecule, it is a carboxyl group. When it's broken off, it's carbon dioxide. So that's our first production of carbon dioxide right there. You can also see that NAD plus becomes NADH. And then the remaining 2 carbons of the pyruvate, it's called an acetyl group, gets attached to something called coenzyme A, which is, it's got a sulfhydryl. Coenzyme A has a sulfhydryl. Man, I think it's this mouse. I'm using a different mouse today. Let's see if I can hide all my ink. So the sulfhydryl group on the coenzyme A helps to link it to the acetyl group, which is what's remaining of the pyruvate. And so if we started with 2 pyruvates, then we just multiply everything on this chart by 2. 2 pyruvates yields a release of 2 carbon dioxides, a production of 2 NADHs, and then 2 acetyl-CoAs are the end product. Again, I'm going to have a summary activity for you to show you all of this. So arsenic, which is a poison, it's an allosteric inhibitor of the enzyme pyruvate dehydrogenase. Remember, I told you pyruvate dehydrogenase is the enzyme that catalyzes this one-step reaction. And arsenic, you probably have heard of it, it's a poison. It's an inhibitor of that enzyme. So if somebody, if arsenic is given to somebody, it's usually in a liquid form, then their respiration stops because their pyruvate dehydrogenase enzymes are blocked. And it stops their respiration, which means they can't transfer energy from glucose to ATP, which means their cells can't do anything and they die. So it's a poison. So just an example of a poison, an inhibitor that relates to this process here. There's a lot of poisons that block enzymes of respiration. For each 3-carbon pyruvate molecule, this is just a review of the last slide, the decarboxylation occurs, and that releases 1 CO2, you get 1 NADH, and you get 1 acetyl-CoA. But then, like I said, there's really 2 pyruvates from each glucose that we began glycolysis with, so you have to multiply all the numbers by 2. All right, the Krebs cycle. The Krebs cycle is 9 steps, 9 enzymes, and this is sometimes called the citric acid cycle because citric acid is one of the first intermediate products that's created in this process. But this is a cycle. This is our first example of a cycle. And in biochemistry, what a cycle means, the word cycle, it means that something that you started with is also going to be one of the products at the very end of the metabolic pathway. So there's 9 steps in the pathway. One thing that you start with is called oxaloacetate, and that name you do need to know. Oxaloacetate. That's going to combine with 1-acetyl-CoA. So 1-acetyl-CoA from the last step, the oxidation of pyruvate. Acetyl-CoA is a product of the last stage, but it becomes a reactant of this stage. Combines with oxaloacetate. Acetyl-CoA has 2 carbons. Oxaloacetate has 4 carbons. So those two things are joined together by a covalent bond, and that becomes a 6-carbon molecule called citrate or citric acid. So that's the first step. So the Krebs cycle is sometimes called the citric acid cycle, sometimes called the TCA cycle, but there's different names for it. Then the citrate is going to be rearranged and decarboxylated. You remember that decarboxylation means removal of a carboxyl group, which means release of carbon dioxide. So there's going to be 2 decarboxylation reactions. So 2 CO2s that are released for each acetyl-CoA that starts the whole process. And then the remaining structure will be regenerated into oxaloacetate. So the 4-carbon molecule that's left behind gets rearranged and becomes oxaloacetate again, which means that the oxaloacetate can then accept another acetyl-CoA and the steps can continue. All right. This occurs in the matrix of the mitochondria. All right. Same thing with, well, the pyruvate dehydrogenase enzyme is embedded in the membrane of the mitochondria, and these enzymes are just floating free in the matrix of the mitochondria. So here's the detailed picture of all the steps. Again, you don't need to learn this now, but biochemistry is coming someday. But anyway, so here is, let me get my pen. So here is the acetyl-CoA. And I like this picture because it tells you how many carbons. Like if you just look at this little part, it's just telling you how many carbons if you just want to look at that. So there's a 2-carbon molecule, acetyl-CoA. It's going to combine with the oxaloacetate, which is already there. That's a 4-carbon molecule. So if you have a 2-carbon and a 4-carbon, if they covalently combine, this goes around the circle clockwise. So citrate is a 6-carbon molecule. And you can see in the detailed structure of citrate, it has several carboxyl groups. So that's where the decarboxylations are going to happen. The carboxyl groups are going to be broken off, at least 2 of them are. So citrate is reorganized to what we call isocitrate, but that's still 6 carbons. Ultimately, the important step here, though, is isocitrate, a CO2 is going to be broken off of that and released. At the same time, an NAD+ becomes an NADH. Now you have a 5-carbon structure. It's called alpha-ketoglutarate. Alpha-ketoglutarate still has 2 carboxyl groups. One of those is going to be broken off. Now you have a 4-carbon structure, succinyl-CoA. Succinyl-CoA then gets reorganized eventually as oxaloacetate because succinyl-CoA is a 4-carbon. So there's a lot of steps actually to reorganize the 4-carbon molecule of succinyl-CoA back to the 4-carbon molecule of oxaloacetate. But when you look at all these steps, just highlighting the molecules that we were looking at, oxaloacetate combines with acetyl-CoA to make citrate. Those you need to know. Citrate is a 6-carbon. Then you have 2 decarboxylations, and you have several NADHs that are formed, and then a reorganization back to oxaloacetate. That's fundamentally what you need to know, and I've summarized that on this slide. Yes, you need to know it. You don't need to know all of the names, just oxaloacetate, acetyl-CoA, citrate, that's it. And then 6-carbon, 5-carbon, 4-carbon, we'll summarize it that way. But you're not going to memorize all the actual structures, but the breaking off of a carboxyl group is what we call decarboxylation. There's 2 decarboxylations that occur, 2 CO2s that are released. And then if you look carefully, there's 3 NADHs that are made. Let's see if I can highlight this. So there's an NADH here, here, and here. And then you have something else over here. You have something called FAD. FAD is another electron carrier. It also picks up 2 electrons, and it picks up 2 protons, so it becomes, when it's reduced, it becomes FADH2. All right, but it's also carrying 2 electrons. So each NAD plus picks up 2 electrons and a proton. Each FAD picks up 2 electrons and 2 protons. We don't care about the protons, though. But so you have a lot of electron carriers that are being produced here. You also have a molecule being formed. You really have what's called a phosphorylation of GDP. GDP is almost exactly the same as ADP, adenosine diphosphate and guanosine diphosphate. Both of them are adenosine and adenine and guanine are both nitrogenous bases. And so GDP plus PI gives you GTP, just like ADP plus PI gives you ATP. So GTP is another kind of energy molecule, just like ATP. We talk a lot about ATP in freshman biology, but GTP is equivalent. So either one of them can be energy molecules. All right, sometimes it's an ATP that's produced, sometimes it's a GTP. We usually summarize it as an ATP just to simplify. All right, but the oxaloacetate is regenerated in the process, so that's the part that keeps circling. We'd say cycling. All right, so here's your summary. For each 1 acetyl-CoA that enters the Krebs cycle, 2 CO2s are produced, 3 NAD pluses are reduced to 3 NADHs, 1 FAD becomes 1 FADH2, and 1 ATP or GTP, but we'll just say ATP, is produced by a direct phosphorylation event. But the oxaloacetate is then regenerated and the cycle can continue on and on. For as many acetyl-CoAs as there are, this cycle can continue to turn with the 9 enzyme steps. All right, so that gets us through the oxidation of glucose. All right, at this point I'm going to take a break and I'm going to insert for you in your playlist an activity where you summarize all of the reactants and products of the important ones that we need to memorize for glycolysis, for pyruvate oxidation, and for the Krebs cycle. So I would recommend you go through that activity, fill out that little worksheet first, and then we'll delve into, I'll give you the lecture on this part of cell respiration, the electron transport chain, chemiosmosis, and oxidative phosphorylation. So I'm going to stop here and let you do that activity before, I would suggest you do it before you continue with the next part of the lecture.